Hello students, let's discuss about post-transcriptional processing in eukaryotes today. In introduction, the flow of genetic information is simple in prokaryotes compared to eukaryotes, as the process of transcription and translation is a simultaneous process and takes place in the cytoplasm of the prokaryotic cell. However, in eukaryotes, the process is separated by the nuclear membrane, which allows transcription in the nucleus and translation in the cytoplasm. Moreover, the transcript produced in eukaryotic nucleus is a raw product called the primary transcript and need to be processed to become a functionally matured molecule. The process of modification of the RNA primary transcript to give it a functional identity is called the RNA processing. RNA processing involves addition of chemicals and modification of bases, as well as removal of certain unwanted sequences present in the primary transcript. Although we refer to RNA processing as an inherent process exclusive to eukaryotes, prokaryotic tRNA and rRNA do undergo modification from their primary transcript to become mature RNAs. In eukaryotes, all three major forms of RNA, that is mRNA, tRNA, and rRNA, undergoes varying degree of modification in the nucleus before being exported to the cytoplasm where they become functional. Processing of mRNA in the eukaryotic nucleus involves modification of its 5' as well as 3' terminus and removal of extra non-coding sequences from the primary transcript. The 5' terminus is kept and the 3' terminus is modified to a 150 to 200 poly-A sequence. We will also discuss about tRNA and rRNA modifications in eukaryotic cell. Let us first discuss about 5' capping in mRNA. In mRNA of eukaryotes, the 5' end is altered to accommodate a methyl guanosine residue in a process called mRNA capping. The mitochondrial and chloroplast mRNA, however, are not capped. The 5' cap consists of a guanine residue covalently linked to the mRNA in an unusual 5' to 5' triphosphate bonding. The guanine residue is then methylated at the 7th position after capping. In the capping process, one of the terminal phosphate group is removed by RNA triphosphatase, resulting in a bisphosphate group. The GTP molecule is then added to the terminal bisphosphate by mRNA guanyl transferase after losing a pyrophosphate from the GTP. This results in a 5' to 5' triphosphate linkage. This process is followed by methylation of the N7 of the terminal guanine residue. In the next process of mRNA processing, we will discuss about 3' polyadenylation. As the RNA polymerase II during transcription reaches the end of the gene, two proteins called cleavage and polyadenylation specificity factor CPSF and cleavage stimulation factor CSTF recognizes the poly-A signal transcribed on the pre-mRNA. This leads to cleavage of RNA and followed by polyadenylation by poly-A polymerase, which adds approximately 200 adenine residues to the 3' end of the RNA without the need of a template. In the next process, we'll discuss about intron splicing. When introns were discovered, it was known that the nucleus contains large number of RNA molecules of varying sizes, which are referred to as heterogeneous nuclear RNA. These RNAs are primary transcript of polymerase II and found to have short lifespan, which is in duration of minutes. It was also observed that most of them are capped at the 5' terminus and polyadenylated at the 3' terminus. It is now well established that the heteronuclear RNA are primary transcript of mRNA. The generally lengthy nature of HNRNA is due to the presence of segments of untranslated regions called the introns. HNRNA population in the nucleus constitutes precursor mRNA at various stages of processing before they finally leave the nucleus. The discovery of introns in the primary transcript influenced the research in finding out the mechanism of how they are removed from the primary transcript and also determining how the sequence is prevented from being expressed as a protein. Finally, it was confirmed that introns were removed during mRNA processing rather than ribosomes skipping the introns during translation. The process of removal of introns and joining of the exons to give rise to a matured mRNA is called RNA splicing. During the splicing mechanism, there is an introduction of breaks at the 5' and 3' end of the introns and the exons. Flanking the introns should be covalently joined. Splicing is a mechanism that occurs in great precision, as the addition or removal of a single nucleotide can be dangerous to the organism due to mutation. In the introns, we have some conserved sequence within the introns. Let us look into it in more detail. In order to understand how the cell machinery discriminates between introns and exons, a large number of sequences were analyzed from diverse species. The analysis revealed that there are conserved sequences that determines the presence of introns in heteronuclear RNA. The most commonly found nucleotide at the exon-intron junction is the GU at 5' splice site and AG at the 3' splice end. The sequence is conserved in a vast majority of pre-mRNA transcripts in eukaryotes. Besides the border sequence, there is a third sequence that is present within the intron, which is essential for splicing. The sequence is called the branch point, which is a 100% conserved adenine nucleotide followed by a tract of polypyrimidine sequences. It is noted that the base constituent of the introns is irrelevant to the mechanism of splicing. Any mutation at the conserved region leads to unspliced introns, which determines the importance in mRNA processing. For example, certain types of disorders like inherited thalassemia are commonly caused by mutation in the splice sites of the globin gene. Now let us consider discussing about chemistry of splicing. Introns can be classified into 4 groups, which are group 1, group 2, group 3, and group 4 introns. Group 1 and group 2 introns are self-splicing introns, whereas group 3 introns require additional proteins and group 4 requires enzymes for splicing. Let us first discuss about group 1 intron splicing mechanism. Group 1 introns are found in a number of genes, including few rRNA genes in protists, mitochondrial and chloroplast genes in plants, as well as fungi, and even few bacteriophage genes. Although the polynucleotide sequence of group 1 introns may be different, collectively they have a conserved secondary structure. Some of the important structural features are a binding site for G nucleotide or nucleoside, and an internal sequence with G residue that base pairs with the 5' splice site. This sequence determines the exact site of cleavage. Self-excision of group 1 introns proceeds through 2 trans-esterification reactions. A free guanine nucleotide or nucleoside binds to an active site on the intron, and the 3' hydroxyl group attacks the 5' end of the intron. The free 3' end of the exon then attacks the 3' splice site, which is the second trans-esterification reaction, resulting in fusion of 2 exons and release of the intron. Though in this case, intron is released in a linear form and there is no formation of a lariat structure. In group 2 intron splicing mechanism, group 2 introns constitute the other class of self-splicing introns. They are present in few mitochondrial and chloroplast genes of plants and fungi, as well as in the genomes of some archaea and bacteria. Like group 1 introns, they are two-fold into characteristic secondary structures. But the chemistry of splicing and RNA intermediates produced is the same as those for spliceosomal-mediated splicing of nuclear genes. They use the adenine residue within the branch site to produce a lariat, and finally, the intron is released in the form of a lariat. Because of these similarities, group 2 introns and nuclear pre-mRNA introns have been suggested to be evolutionarily related. Perhaps the nuclear pre-mRNA introns evolved from the self-splicing group 2 introns, and later snRNAs of the spliceosome took over the catalytic function. Removal of the introns requires cuts at both splice sites so that flanking exons become free to join each other. This process takes place through the trans-esterification reactions. In the first trans-esterification reaction, the 2' hydroxyl of adenine residue in the branch site attacks the 5' splice site, and the ester bond between the sugar of the last nucleotide of 5' exon and phosphate of conserved G at the 5' splice site is broken. The free 5' phosphate of intron now forms a new ester bond with the 2' hydroxyl of the conserved adenine at the branch point site. This results in a three-way junction at branch site adenine. This reaction also frees the 5' exon from the intron. In the second trans-esterification reaction, the 3' hydroxyl of 5' exon attacks the 3' splice site. The ester bond is broken at the 3' splice site, and simultaneously a new phosphodiester linkage is formed between 3' hydroxyl of 5' exon and the 5' phosphate of 3' exon. As a consequence, the two exons are covalently joined together and the intron is released. Because the 5' end of the intron had been joined to the branch point of adenine in the first trans-esterification reaction, the newly liberated intron has the shape of a lariat. In group 3 intron splicing mechanism, the splicing reaction described above are mediated by a huge molecular machinery called spliceosome, comprising of RNA and proteins. Spliceosomes are in many ways like small ribosomes. Each spliceosome comprises of 5 small nuclear RNAs, also referred to as snRNAs, and about 150 proteins. Because they are rich in uridine, these snRNAs are named as U1, U2, U4, U5, and U6. U3 snRNA is found in the nucleolus and is possibly involved in the processing of ribosomal RNA. These snRNAs, which range in size from 100 to 300 nucleotide, in most eukaryotes are complexed with several protein molecules to form small nuclear ribonucleoprotein particles, also referred to as snRNPs. Sometimes we call it SNRPs. The U1, U2, and U5 snRNPs each contain a single snRNA molecule, whereas U4 and U6 sRNAs are complexed to each other in a single snRNP. Spliceosomes are not present within the nucleus in a prefabricated state. Rather, they are assembled in a stepwise manner as their component snRNPs bind to pre-mRNA. Once the spliceosome machinery is assembled, the snRNPs carry out the reactions that cut the intron out of the transcript and paste the exons together. There are also many proteins within the spliceosome which are not components of snRNP but play critical roles in spliceosome assembly, particularly in identification of correct splice sites in pre-mRNAs. Introns frequently contain many sequences that resemble splice site, so the splicing machinery must be able to identify the appropriate 5' and the 3' splice site at the intron exon boundaries to produce a functional mRNA. These proteins bind to specific RNA sequences and then recruit snRNPs to the appropriate site on the intron. One example is the U2AF which recognizes and binds to the polypyridine tract at 3' splice site and recruits U2S RNA to branch site. There is another class of protein called SR proteins, rich in amino acid serine and arginine, which binds to the specific sequences within exons and recruit U1S NRNP to 5' splice site and U2AF to 3' splice site. SR proteins, along with many other components of spliceosome, become associated with C-terminal domain of RNA polymerase II as transcription begins. This anchoring of the splicing machinery to RNA polymerase ensures that exons are joined in the correct order and no exon is skipped during mRNA processing. The majority of pre-mRNA in higher eukaryotes are spliced using this splicing machinery. After we learn about the splicing, let us learn in detail about RNA editing. RNA editing is another kind of post-transcriptional modification that can alter the protein coding sequence of mRNA. It was first discovered in mitochondrial mRNA of trypanosoma, in which the U residues are inserted and deleted at multiple sites in the mRNA prior to translation. As a result, the ribonucleotide sequence of the mature RNA differs from the sequence encoded in the exons of the DNA from which it was transcribed. Since the time of its discovery, several examples of RNA editing have been observed in mitochondrial mRNAs of other organisms, chloroplast mRNA of higher plants, and nuclear mRNA of some mammalian genes. There are two mechanisms by which editing occurs in mRNA. The first one is by changing the structure of individual nucleotide bases, and second mechanism is by inserting or deleting nucleotides at specific sites in the RNA. Next, let us learn about post-transcriptional processing of tRNA. The tRNA molecules in both bacteria and eukaryotic cells are transcribed as precursor tRNAs that are extensively modified and processed to produce mature tRNAs. In prokaryotes, several tRNAs are usually transcribed together as a one large precursor RNA, which is then cleaved into pieces, each containing a single tRNA. Some tRNAs in bacteria are cleaved from the large precursor rRNA. In eukaryotes, tRNA genes are clustered, but RNA polymerase III transcribes each gene individually rather than as a large precursor. Processing of tRNA typically involves removal, addition, and modification of nucleotides. Pre-tRNAs are generally longer and mature tRNAs and have additional nucleotides at 5' and 3' ends called the 5' leader sequence and a 3' trailer sequence respectively. These sequences are removed by specific enzymes. The 5' leader sequence is cleaved by an enzyme called RNase-P, which is a special endonuclease consisting of both RNA and protein subunits. In 1983, Sidney Altman and his colleague demonstrated that RNase-P is a ribozyme. Each RNA subunit can catalyze the cleavage of pre-tRNA independently of the protein subunit, though both are required for maximal activity. The 3' trailer sequence is cleaved by a conventional protein RNase. All tRNAs have a trinucleotide sequence CCA at their 3' end. This sequence is the site of amino acid attachment and is essential for tRNA function during protein synthesis. A special enzyme called CCA adding enzyme adds this sequence post-transcriptionally at the 3' end of all those tRNAs that lack this sequence in their primary transcript. Some tRNAs already have CCA in their primary transcript and therefore do not require this addition. Some pre-tRNAs in a few organisms contains introns of variable length that are removed by splicing. The mechanism of intron removal from the pre-tRNA has been discussed in the earlier section on splicing mechanisms. Another important aspect of tRNA processing is the extensive chemical modification of a number of nucleotides by specific enzymes at specific position in the tRNA. Base-modifying enzymes change some of the standard bases like U, C, A, and G of the pre-tRNA into modified bases. The type and extent of this modification vary from one tRNA to the another, but typically include methylation of bases and sugars and creation of unusual bases such as dihydrouracil, pseudouracil, ribothymine, and inosine. A typical tRNA molecule has about 10 to 15% of the nucleotides modified during processing. Some nucleotide modifications are found at the same position in nearly all tRNAs. For example, pseudouridine is found at the same position in T-Cy arm in all tRNAs studied. Other modifications may be unique to one or a few specific tRNAs. The exact function of most of these modified bases is unknown, but it is believed that their presence assists in forming a particular three-dimensional configuration of the tRNA, which in turn determines the function of these molecules, specifically their ability to pick up a specific amino acid and bind it to the ribosomes. Modification in the anticodon loop of tRNA makes the wobble effect possible. In conclusion, the eukaryotic mRNA is a product of extensive modification of hnRNA that is synthesized in the nucleus. The process of transcription is simultaneously followed by modification of the primary transcript to yield a functional form of RNA. All the three major forms of RNA, which are mRNA, tRNA, and rRNA, undergoes stages of modification before finally being exported out of the nucleus. In the case of eukaryotic mRNA, the process of alternate splicing is made possible due to the association of snRNPs at the intron splice sites, which allows selection of introns to be cleaved unlike the predetermined ligation of exons in a series.